Okay, today we're going to do the the extracellular matrix. And we're also going to do cellular junctions. In the movie E.T. with the creepy little alien, hate that movie. What does E.T. stand for? Ryan, what does E.T. stand for? Extraterrestrial. Reagan, what does terrestrial mean?
Like of Earth. Yeah,
of the Earth. If there's terrestrial life forms, there's marine life forms, there's airborne life forms, aerial. What does extraterrestrial mean? Jasmine? Out of this world, beyond the Earth, beyond the land. So extracellular means beyond the cell, outside of the cell. So let's talk about this extracellular matrix thing. A matrix is typically a network of like interwoven processes or interwoven structures. An extracellular matrix is known as the ECM, and you only find it in animals. I'll explain why momentarily. The extracellular matrix is not just a void. It's not like outer space. There's actual stuff that makes up the matrix. It's, like I said, it's like a big, vast network of interwoven structures and processes. This is a series of proteins. Man, there we go. A series of proteins that gives animals structure and holds cells together. Now, the reason why you're not going to see this in plant cells is what do plant cells have on the outside of their plasma membrane that our cells do not? Cell wall. So take a look at how closely packed they are to each other. So these are plant cells. There's really no space between them. Plants are very densely packed. Their cell walls make it very rigid. Not a lot of space there for an extracellular space, much less a matrix. But when you look at animal cells, they're much more pliable, they're much more durable. And if you look closely, many of your different types of tissue have spaces between the cells. It might be a very narrow space, but a space nonetheless. So we're going to talk about this void, and what's in there that keeps these cells in place. Because the cells don't all just bounce around, they're held together. So there's going to be a few words here I'm going to introduce to you guys, and I'm going to introduce the terms to you, I'm going to define them, and then I'm going to make what I think is going to be an awesome illustration to really bring it all together so that you can see just how all these animal cells are actually but holding to each other. So the first term is called integrin. Integrin is transmembrane proteins, and just by hearing that term, you've had this earlier. What does it mean to be a transmembrane protein? They go through the plasma membrane from one side to the other. So transmembrane proteins that attach on the inside of the cell to the microfilament mesh. And microfilaments are part of what major network within the cell? Cytoskeleton. The cytoskeleton is made up of microfilaments, intermediate filaments, and microtubules. It's what gives the cell its shape and support and reinforcement. If you were to take your bones and your skeleton out of your body, you would turn it into a big pile of goo. Same thing, if you know, here's my elbow. If I'm pushing and my finger stops because I have bone there. If I didn't have any bone there, my finger would go all the way through my arm. Well, the same logic goes with cells. They have to have a reinforcement to keep the membrane up and to keep the organelles in place. So we are going to be using the word cytoskeleton here when I do my drawing, because I have to draw the microfilaments. Okay, the next term, funky term, is going to be fibronectin. So fibronectin attaches to the outside of an integrin and anchors it to the extracellular matrix. So we just did integrin, okay? Imagine that you're tying up your boat to a dock. The dock and the cleats, you know, the thing you wrap the rope around, that's the integrin, guys. The fibronectin is your rope. So what the heck is this fibronectin attached to? A boat. There are no boats in the cell. This is just an analogy. But we're about to go over what the quote-unquote boat would be. This last one is going to be proteoglycans. Proteoglycans are a class of protein that forms the structure of the ECM. Now, these are just the definitions, and what I'm going to do is I'm going to draw an image for you. But if I were to make this an analogy of a marina, actually, I'm going to flip something here. The boat is a cell. The rope is the fibronectin. The integrin is what you tie the rope to on your boat. A lot of boats have cleats or they have posts where you wrap a rope around to tie it up on a dock. The docks are the proteoglycans. So let me just do this really crude drawing here. If I have a boat, there's the motor, blah, 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 blah. And here's the dock. You know, here's another dock. Here's another dock. The boat's going to be the cell. Okay? You don't need to draw any of this. You can draw the real image. The ropes in which you're tying the dock to the boat, those are going to be the fibronectin. The boat, or excuse me, the dock is going to be the proteoglycan. In the actual parts of the boat in which the blue things right here, the fibronectin attached to, those are going to be the integrins. So enough boats and docks and marinas. Let's actually put this to biology. So I'm going to draw two cells. So the black lines represent the plasma membrane of a cell. Now within the cell, you have actin filaments or microfilaments. They're the same thing. These are the cytoskeleton. Without this, the cell is just going to collapse on itself because it has no reinforcement for the membrane. So this is all just like the skeleton of the cell. It holds it up. It's like the superstructure of a building. Okay, next. You're going to have those transmembrane proteins that go all the way through, and they attach to the actin filaments that are that are woven throughout the cell, especially the ones that are just below the plasma membrane. These yellow structures, which are the proteins that are cutting through the plasma membrane, this is going to be the integrin. It's really hard to see yellow, so I have an improvisation here. I'm going to try to make it gold. Okay, now I'm going to skip fibronectin. I'm going to go straight to proteoglycans. I'll come back to fibronectin. So proteoglycans are going to be proteins that are woven in the extracellular space. It's like a spider web, but the proteins are forming this web, this network. A student in first period asked, where are these proteins made? Well, what is it that I say? When I say ribosome, you say? Right. The proteins are made within the cell, and then the cell gets rid of the proteins. By what process does a cell expel a protein into the extracellular space? Exocytosis. Terrific job. Exocytosis. So these proteins are supposed to be out of the cell, and they're going to go perform some job out there. In this case, they're going to form the proteoglycans. And then finally, now we'll go back to the fibronectin. The fibronectin are the ropes connecting the boat to the dock. In this case, connecting the cell to the extracellular matrix.
Do they connect on the integrin?
They connect what is it? The fibronectins connect the integrin on one side to the proteoglycan on the other. And what you have here, guys, is cells that are stabilized in a region. They're not just going to float around like they're a beach ball on the pool. And this also helps contribute to make our next topic, which is tissues. Okay. our last topic is going to be tissues. By definition, tissues are made up of one or more layers of cells with a common function. Now, in biology, there's a hierarchy. You know, if you get a lot of this, you make that. If you get a lot of that, you make those. If you get a lot of those, you make them. If you get a lot of them, you make these. So let's see if you guys can continue my little hierarchy here. I'm going to start with atoms. When you get a lot of atoms together, what do you make? Molecules. When you get a lot of molecules together, what do you make? Macromolecules. When you get a lot of macromolecules together, what do you make? Organelles. Keep it going. A lot of organelles. What's next? Cells. A lot of cells, what? Tissues. A lot of tissues, what do you get? Organs. Multiple layers of tissue. Oops, I don't know why I just did two. Multiple organs, what's next? Correct. Do you know how many organ systems you have? 11. And then multiple organ systems? Organism. And then you could go into ecology. You could start doing multiple organisms as a population, multiple populations as a community. Then the communities, the ecosystem, ecosystem to biome, biome to biosphere, then planet, then solar system, the galaxy, then universe, and blah, 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 blah. But we're just going to focus on the tissue part. So the definition of tissues is at least one layer of cells with a common function. In order to get them together, you got to talk about the cell junctions, where one cell meets another. So cellular junctions. Cellular junctions are different types of proteins that connect cells. Now, there's a few types of cellular junctions and the proteins they're in. First one is going to be called adhesion junctions. Adhesion junctions are kind of like the parts of a jacket that snap together to close the jacket. These are going to be proteins that hold cells together. The strongest type of adhesion junction, this is a fun word to say, desmosomes. You're going to see that word a lot. Here's what it is. Desmosomes are adhesion junction proteins that provide a strong hold and the ability to stretch without tearing. So Shane, if you were to hold up your arm, you pinch your arm, why don't you go and do this? Pinch the skin of your arm and twist. Don't kill yourself. Now, let go. What happened to your arm? It went right back. Because you obviously have a lot of elastin and collagen, but our skin, our tissues are very tough. Unless you get a knife and open your arm open your skin open, you're very durable. However, you guys are like 2/3 water. Your blood has water, your cerebrospinal fluid has water, your cytoplasm has water. There's a lot of water everywhere. Why don't we leak? Because the little, the handicap with these is that they are not watertight. So they're good for pulling and stretching and not tearing, but they're not good for retaining water. So you have to have another type of adhesion junction that retains water. And those are called tight junctions. Tight junctions are watertight. They hold cells very close together. These two work together in our tissue. And not just our tissue, but most animals. Definitely mammals. And I will draw these in about 2 minutes. I'm going to draw this all because I know that a lot of you are visual learners, and a picture goes it speaks 1,000 words, way more than words do. So the last part for today, before I draw you your pictures, are another way in which cells connect to each other. This is going to be called a gap junction for animals. And plasmodesmata for plants. Now, both of these do the same thing, but the reason that you have two different types is because what do plant cells have that animal cells don't have? Cell wall, amongst other things, but the cell wall is what I'm focusing on here. Despite the fact that you have two different ones, they both allow for the transfer of ions, water, and nutrients directly between cells. So back there in the back of the room, behind Annie, is a door leading to Miss Smith's room. That could be my little gap junction or plasmodesmata with her classroom. It's just a connection. So to end today's class, I'm going to draw you guys several types of cells. Same types of cells, but with different types of junction proteins, okay? So these are all just cells. And I'm going to zoom in on this region right here. I'm going to draw you guys a desosome. So right at the membrane, you have the place where they're going to attach. They form a bridge. It's a very pliable bridge. It's a pliable junction that really resists tearing and shredding. Over here, I'll draw some tight junctions. It's like stitches. And they really do bring the cells very close together so that there's no water loss. It's like stitches from a doctor. Okay, and the last thing, if you just give me like 10 seconds, I'll get this out. This right here is a gap junction. It's just a tunnel for the cells to be able to communicate with each other. The same goes for plasmodesmata. They're just little tunnels connecting one cell to another, to another, to another. All right. That's it.